Okay, so I'm going to talk about some of the chemistry functionality that's new in version 13.1, which is pattern reaction and apply reaction. We've had molecules in the Wolfram language since version 12, and I will touch on that briefly in case you don't have much background in that, so you can follow along with the rest of the presentation. I'm going to be talking about chemical reaction networks, where you essentially, instead of doing just a reaction on some reactants and you get some products and you stop there, those products can become reactants for the same reaction or for other reactions that are available, and you end up generating a series of products. And what's interesting about this is that sometimes you can get to one of those products downstream by a number of different routes through the network. And it becomes a multi-way system in a sense. And so we've been looking at that aspect of it as well. Okay, so let's get started. You've all read my abstract. These are the things I'm going to cover. In the chain reaction section, I'm going to skip over the Diels-Alder reaction just so we can get to the end today, but it's in the notebook. It's all there. You can run it without a problem. On the rearrangements, I'm also going to skip over the Cope rearrangement, although it's very interesting. There won't be time to cover everything. I'm going to jump, after the introduction, I'm going to jump straight down to the Wagner-Meerwein rearrangement. We're going to talk about carbocation rearrangements and then how those are used in plant metabolism in terpene biosynthesis. This is what I did my graduate work on 40 years ago. And I've been wanting to be able to do this sort of work computationally ever since then. And now that we've got pattern reaction and apply reaction, I can actually do some of this work. And that's what the bottom line of this presentation is about, is getting to the point to do that. Okay, so reaction networks, we've seen a lot of them in biology. Many of you have seen these charts like this. I can zoom in here, and this is the tricarboxylic acid cycle, or the Krebs cycle. And there are different molecules in here and the enzymes that convert one to another. And we can see, for example, here, oxaloacetate, while it's involved in this cycle here, it also can come in from a number of different sources in your cellular metabolism. And so it's a real multi-way system. If fatty acids are produced in a repetitive manner, an iterative manner, you start out with acetylacetate here, and you add 2 carbon units to it as the chain increases in length, and eventually you come out with a 16 or 18 carbon long fatty acid. And again, you can think of this as a reaction network. In organic synthesis, we also have networks. Synthetic chemists will start out with a pair of reactants, and they will glue them together, as it were, and then add more material to it. Oftentimes, the synthetic pathways are quite linear. This is Woodward's synthesis of strychnine. It was 28 steps in all, getting to the final product here, but it was done in a very linear fashion. Others are convergent. This is Corey's synthesis of aspartidophytene, and he did it in 3 steps. He built the first part in a linear fashion, this molecule here, and then he built the second part, this one, again in a linear fashion, but then he took both of those, converged at a single point then, and then through a number of steps reached the final product. What I'm going to be talking about today, though, are going to be rearrangements, mostly the Wagner-Meerwein one. This is an example here of where it was first discovered, essentially taking the borneol molecule here and removing under acidic conditions the hydroxyl group of the alcohol. It leaves a positive charge on the carbon. And then the bonds in the structure can rearrange. This bond here shifts over to this position as shown here. And if you redraw the structure, it has this shape. It looks like the 2 methyl groups have shifted over here and this methyl group has shifted there, but really It's this bond, this one here, that shifted to do that. These are the kind of rearrangements that the enzymes in plants carry out on these terpene compounds to make a very wide variety of them. For example, things, this one is borneol, a related one is camphor. It's been an ingredient in mothballs, for example. Things like menthol as flavorings. A lot of your perfumes are made up of monoterpenes and sesquiterpenes, but they're very volatile and they produce scents. Why do plants produce these? I don't know for sure. A lot of them, they could be attractants to attract insects to pollinate the plant. Some of them could be to repel insects from eating the leaves and things like that. They're all part of the secondary metabolism, and they're just very wide variety of structures, and that's why chemists are interested in them. So in chemistry, in the Wolfram language, we're dealing with models. And we've got models for the structure, and it's the atoms and the bonds and their arrangement in space. And we have models for reactions, and that's how we can change the arrangement of the atoms within the molecules. For example, we can take this aldehyde molecule here, acetal, and reduce it with sodium borohydride to the alcohol. And the way we do that in Wolfram language, like I said, is a model. And as you may have heard in an earlier talk this morning, all models are wrong, but some are useful. Under the hood, we're using graphs to represent the molecules. The atoms are vertices, the bonds are the edges of the graph. And we're going to use graph rewriting to carry out reactions. In other words, rearrange the edges among the vertices to represent a reaction. So molecules in Wolfram language are built with a molecule function. Okay, so here's that same molecule, citronellal. And the reaction we want to do is to reduce it with sodium borohydride. Here's the sodium, here's the borohydride. And the graph rewriting is what organic chemists call arrow pushing. And so we're pushing the bonds or the edges into new locations, and we produce products. And now we've got a new bond here and a new bond there. We've lost that one and we've lost this one. And this is now the alcohol. This is the borane. And now we've got sodium methoxide as the product instead of the sodium borohydride as the reactant. How do we carry this out? We use pattern reaction. Pattern reaction takes a couple of different kinds of input. One of them is the SMARTS notation, which a lot of computational chemists understand. Basically, you define a set of reactants on one side of an arrow and your products on the right-hand side. And you also have to label the atoms with numbers so that we know which atoms, where they start, and then where they end up. And so we can just define the reaction this way. We get a much better depiction. It would be great if we could just draw them in this way. But we can see here that this oxygen starts out here in this reactant and ends up over here in the alcohol. Here is the oxygen of the methanol, ending up over here is the oxygen of methoxide. The graph rewriting is carried out with the apply reaction function. And so let me define the reactants here, the sodium borohydride and the methanol. We've already defined the citronellal. We can carry out the reaction with apply reaction here. And we get the three products. And I can plot them pictorially for you. So here's the alcohol plus the borane plus the sodium methoxide. So this is basically how we do our chemistry in Wolfram language. Okay, now we also have models for reaction networks, and these are multi-way systems. We generate the multi-way systems is an iterative process. Some of them terminate, in other words, they get to a fixed point and they don't change anymore. Others could be infinite and just keep going on forever and ever. And chemical reactions can be like that as well. The reactions, though, will tend to terminate mostly because you just can't keep adding stuff to make things infinitely big. Polymers get very large, but even then in polymerizations, there's usually an upper limit for the length of the polymers. Okay. So we can use the multi-way system. You've probably seen these string rewriting systems before. Stephen has been using them in the physics project. And we can do the same sort of multi-way system using the token event graph function. The token event graph function is more general. You get to specify the rules you need to use as well as what they work on. So you've got much more control over the system you want to write, that you want to study. So I can do the same multi-way system. I've got the same rules here for the strings and the same initial state A, but here I have to specify what carries out the transformation string replace list, which is built in to the multi-way system function here. Because I have to specify it here, I can swap this out and put in ApplyReaction, for example, and that's the whole key for it. So just a quick demonstration, I get the same multi-way system here as the token event graph. The tokens are the strings here, and the events are the yellow circles of the different applications of the rewriting rules. In the chemistry sense, we'll do the same thing. The tokens will be the molecules, and the events will be the reactions. So just to demonstrate that, we can do a very simple polymerization right here to make polyvinyl chloride or PVC. It starts out with a homogenization reaction of a peroxide to form a radical. That radical then reacts with vinyl chloride to produce a radical at this end of the molecule now, which is here in the reactant, and then we can elongate that by repeating it. This radical reacts with a vinyl chloride to make it longer and still produces another radical, and that's what lets us have the chain reaction and so on. So we define our patterns for the reaction and the reaction itself, the elongation, and then we can define some starting materials here. And we can just do this with nest list 5 times, and we start out with the oxygen radical here, and then we get to the first step, second, third, fourth, and fifth. And then I can do the same thing with token event graph. I'm using the same elongation reaction. I'm using the same vinyl chloride. I'm starting with the alkoxide radical. We're going to go 5 steps. And I'm going to use the molecule plot function to render the molecules. So we don't see them as the summary box, but we see them as a molecule plot output. And so this is the token event graph for that. And we can see here, we start out with the alkoxide radical, and then the first chloride radical, the next, and there's the elongation. And this is a token event graph. And each of these yellow circles here represent a specific reaction event. Okay, this system that we're using is a model, okay? There's no energetics, no geometry in here, no stereochemistry, nothing like that. It's really just graph rewriting, but it serves our purpose to study the gross nature of these reactions. Also, on the flip side, this is, token event graphs are a mathematical construct. And we don't want to get, as chemists, I don't want to get caught up in the mathematical aspects of it and lose sight of the chemistry. And so I'm focusing more on the chemistry side today.
When you look at the events, when you look at the events, okay, so the yellow circles?
Yeah.
Okay, can you dive down into it?
Yes, I will show you how we can pull those out and find out exactly which the rules were applied at those different steps. Yep, that token event graph keeps track of all of that. You can tell it to some of the visualization options to tell it which rule was actually used in a given event. Okay, I'm going to skip over the Diels Alder so that we can get down to the nuts and bolts of the plant biosynthesis with the terpenes. So for the Wagner Meerwein, I showed you the Borneol molecule where the reaction was discovered. It's based on carbocations. These are molecules that have carbons in them that bear a positive charge. There are 5 different kinds of them. These are the names: primary, methyl, primary, secondary, or tertiary. And it depends on how many carbons are attached to the carbon that bears the plus charge. And the more carbons that are attached, the more stable it is. And that's because the carbon atoms can donate electrons toward the carbon that bears the charge, which stabilizes it. So things like primary carbocations will tend to rearrange very fastly to form a secondary one. And secondary ones will tend to rearrange to form tertiary ones. Tertiary ones can go back to secondary, but it's very rare for a secondary carbocation to go to a primary one. Okay. So here's an example of doing a couple of migrations in a row. Hydrogen migration. One of these hydrogens migrates here, and there's the hydrogen that's migrated, and now the charge is here. And now I can migrate the methyl group here over to here, and the charge goes back. And then I can migrate this hydrogen there, and now I've got 2 hydrogens here, and the charge is over at this end. Okay, so because there is some energetic preferences for the types of rearrangements that one can write, and I want to take that into account because I don't want to try to model, to simulate every possible type of rearrangement, but only the ones that are most likely to occur in plants. I'm writing very specific reactions for each of the different kinds of transformations, and I am specifically leaving out those that will produce a primary carbocation. So I've written them here, and I've just iconized the definition here just to save space, but pictorially we can see what they look like, how the atoms move from reactants and where they appear in the products. We can do the same for the hydride shifts. We've got a set of those. And to demonstrate this, I can start out with this isopentane molecule with a charge on it. So it's the isopentane cation. I've labeled the atoms here with numbers so you can watch them, where they end up in the products. The reason I'm doing this is with molecule plot, I don't have a lot of control if it's going to draw the molecule this way on the page, or if it's going to turn it around that way or flip it. So I'm numbering things so that you can keep track of it a little bit better. So notice that the charge here is on number 2. That's where we're starting. And we're going to use a hydride shift followed by an alkyl shift followed by another hydride shift. And so we can carry out those 3 reactions. We start out with a charge here. The first thing we do is a hydride shift. So we're shifting a hydrogen from 4 to 2. 4 gets the charge. 2 now has a hydrogen on it in addition to the 2 carbons. We do an alkyl shift where carbon 1 is now moving over to carbon 4. Carbon 1 is now on carbon 4, although it looks like it hasn't moved. And then we do another hydride shift that shifts the hydrogen that's not shown here on 4 onto carbon 2, and the charge ends up there. Okay. So we can do the same thing with the norbornyl cation. This is the same thing as we saw before with borneol. It's called norborneol because we've removed the 3 methyl groups. And so it's just the 7 carbons of the 2 rings. And if we look at it in 3D, this gives a little bit better idea of what the structure is that we're dealing with here. And this is the carbon that has the charge on it. This is the 1 carbon bridge that's the middle, that's C5. Here's C2, the one with a charge. Okay, so there are two different ways we could do alkyl shifts on this molecule. We could shift this bond here from the 1 carbon bridge over to here, or we could shift this bond here underneath over to here. This is the one in the first example I showed on the earlier slide is the one that shifted. So when we do this, because there are 2 possible ways we can do it, And we've asked ApplyReaction to do all possible ways. We end up with the two different products. One of them, the shift of this one underneath, did not change the overall shape of the molecule, but if you follow the labels, you'll see that things have actually rearranged. So to do the token event graphs for the full multi-way system, we define our rules, which is taking all of our alkyl shifts and hydride shifts. We have 14 rules now. And I'm going to tell token event graph to color the event vertices with a different color and number representing which rule was used. And I'm only going to go one step. And when we do that, here's where we start. And the pink atom here is the one that has the charge. The alkyl shift reaction that gets applied is reaction number 4 in our list. And we can see we get the two different products I just showed you before, and we can see where the carbocation ends up. And there are two different hydride shifts, one that shifts the hydrogen from the C1 to here. That gives us this product, that's this one, and one that shifts a hydrogen from C3 to C2, and that gives us this product here, putting the charge on C3.
Is there a higher probability of one reaction?
Yes, there would be energetic differences and geometry differences that would make one be more favorable, more stable. The more stable one might be produced more slowly, though. There are kinetic effects as well as thermodynamic effects that can be taken into account. I'm ignoring all of that today. Okay, this is just to get the lay of the landscape of what could possibly happen. And once you get biology involved with enzymes, the rules, you know, those relative energies and stuff and rates can all change. And in fact, that's what's fascinating is that the enzymes in different species of plants sometimes do things one way or do things another way. And that's what provides a great variety. And that's why biologists and chemists are very interested in these sorts of things. Okay, I ran this reaction, set of reactions, and after 9 steps, the token event graph stopped changing. It didn't add any more new tokens to it and have any more new events. And this is what the final graph looks like. The starting material for this one is the norbornyl cation, which was here, and it rearranges in lots of different ways. And you know, for example, I can say, you were asking, you know, how can I find out which reactions were used in a certain pathway? So if I want to find out how did I get to this particular molecule here in the network from the starting one, I can actually ask it to find the shortest path on that graph. And, oh yeah, okay. And then I can show what it looks like here, and I can label it with the names of the specific reactions for that. So we start out here with the secondary cation, and we get a rearrangement where this bond moves first, okay, and we end up with this 4-membered ring instead of a 5-membered ring. Then we do a hydride shift, shifting a hydrogen from here to here to get the plus charge there, and then we do another hydride shift, shifting a hydrogen from here to here or there to there, depending on your perspective. And then finally, another carbon shift, shifting one of either this one or that one to this position to give us this 4-membered and 5-membered ring compound. So we can actually dive in and find out what's happening in the network and relate it back to chemistry. We could redraw the same token event graph this way. And here we've got the starting cation at the top and a cascade of reactions that go down. All of these structures here, these carbon skeletons, are found in natural products, and these molecules have been isolated from plants. The ones below the dashed line haven't been found in plants, okay? Which is, I find very interesting. There's a nice clean cut here. Another thing that's useful to look at here is that this is a multi-way system. There's more than one way that I could get to that molecule. I could come down this way here, like this, and get there. Or I could come down this way, which is the route that I had just shown you, to get there. I could also come down this way, down to here, and then back up. So what's nice about this method is it shows you that there are a number of different possibilities that a given particular molecule could have been formed by the plants. And so it's the challenge now is to the chemists and biologists is to figure out how that actually happens. And there are ways they can do that. They can start out by building this molecule with a different type of carbon or hydrogen in there, a labeled one, something like a tritium atom that's radioactive, and you can see where that ends up in the product, or with something like deuterium, which is a heavy hydrogen isotope, and you can see that spectroscopically. You can label it with carbon-13 and see that in C13 NMR spectrum and follow where the label goes. And that's actually what I did in graduate school, in a large number of experiments on a molecule like this, seeing where the labels start out, where they end up, and then inferring which pathway was followed by, in my case, a fungus that produced the metabolite. Okay.
Just a quick question. Does the functionality display the work like this?
I just changed the graph layout.
Okay.
I just changed the graph layout from the default, which is a spring electrical embedding, to the layered directed acyclic graph layout. And I didn't even have to tell it that this one was the root. Oops. Trouble with big notebooks, they scroll slowly. I didn't even have to tell it this one was the root, but I could have. The code for this is here to see what I did. Okay. It is the layered directed acyclic graph layout will take cyclic graphs as well and lay them out nicely. And it only looks cyclic this way because I've got reactions that go in both directions. I did not write my rules reversibly. It just turns out I've got pairs of rules that go in opposite directions when they get applied to a specific molecule. Okay, so getting on to the metabolism then, we start out to make these terpene molecules, these C5, 10, 15, 20, 30 atom molecules that also include the steroids. We start out with a 5-carbon molecule here, and we add this slightly different 5-carbon molecule to it. We get a 10-carbon one, we add another unit, and we get a 15-carbon, add another 5-carbon unit, we get a 20-carbon. This is essentially a polymerization reaction, just like the one we saw earlier with the vinyl chloride polymerization. When I was in graduate school, Devin and Scott had just published a book, it was a several volume book on natural products, and they had in there a chart showing how these terpenes could be made starting from a linear molecule and by different ways of cyclization and rearrangements, getting to all these different natural products. And I thought this was really cool. And I said, we should be able to do this with a computer. And now we can. For the 15-carbon molecules, we actually have 2 pages worth of rearrangements. He only had 1 page for the 20-carbon molecules. There's probably really more. And one thing I hope to be able to do is to use this technology to explore the sesquiterpenes, the C15s, and the diterpenes, the C20s, and fill this out more. So our strategy, we'll start out with these alkyl shift and hydride shift reactions. We'll add 2 more. We need something to close rings here, which is really just an alkylation of a double bond. The plus charge here of the cation reacts with this double bond here, it can attach here and form a six-membered ring, or it could attach at the other end and form a seven-membered ring. Other types of rearrangements that occur if you've got a cation next to a double bond, the double bond can shift over here and move the charge there. And this is called a vinylogous cation rearrangement. So I've added these two reactions to the set. And now we are going to have more reactions to work with. And excuse me, I'm defining this reaction here for the alkylation of the double bonds, and I'm defining this one here for the vinylogous shift. And don't know why it jumped the page, but it did. This is demonstrating the alkylation reaction forming either the six-membered ring if I attach here, or the seven-membered ring if I attach there. And then showing the shift. This is defining the shift reaction. And again, I'm writing several versions of it so I can control which ones actually occur, because I don't want to form primary carbocations in this rearrangement. So I've left that specific one out. And then we can demonstrate it here. We're starting out with a primary carbocation, the double bond shifts, and now the charge is here at this carbon, and the double bond is between the first two atoms. Okay, we can make the token event graph. So I'll define all the rules here. So we had the original 14. I've added 8 more, and we'll define the geranyl carbocation, this one here, which we've seen already. And I'll just show the first two steps. And we have quite a variety of different molecules already made. Some of them cyclic, some of them still linear. Some of them that you'll probably recognize. We've seen this one before. Here's the boryl cation that we saw early on. And of course, these can keep rearranging further down. If I go 6 steps, I generate more than 1,000 different molecules, which is way too big to show. 76 of those molecules, either the molecule itself, the particular cation, was in Devis and Scott's page, his chart, or the carbon skeleton was. The green ones were actually present in this multi-way graph, and the yellow ones, the carbon skeleton was present. If I go those 6 steps, I'm not going to show the whole graph here, but these are the ones that were present in their chart. And these are all of the ways that they're interconnected by this reaction network. There are a couple over here that he didn't include, but they are obligate to get from this one here, for example, to get over to here. So that's why they're included. Okay. And then to dive in, we can look, let's say we want to find this molecule in the set. And we can find out how it got there, that this particular molecule is represented by these 6 different carbocations, the charges in different places in them. I can get the roots out by finding the shortest path. And we have 6 different paths. Here's the first one. And then graphically, I can show 2 of them. Several of them were just different variations on the same theme. So I can get to this molecule here from the starting material essentially in one way through these steps here. Or I could get there through this 5-step process. This one here, based on various experiments with plants, is the one that's actually observed to form this isocanthane structure from the geranyl starting material. This one here, which is a bunch of shifts with finally an alkylation at the end, is not very likely the one that's happening, because no labeling experiments have turned this one up as a possible route. We can look at the formation of this molecule here. And again, it's produced a number of different ways. And here's the first route here, which is rather long. Our model says that we produce it by first forming this ring, and then this forms another ring here to form this one, and then some shifts, and we finally end up with a structure here. They've actually done labeling experiments for this one, and it turns out that this is not the pathway that's followed, but it first forms this structure here, but then the rearrangements that occur based on labeling with tritium to find out how the atoms are actually moving around. They've hypothesized that there are some nucleophiles. These are parts of the enzyme that like a positive charge or double bonds, and they come in here and let the electrons then grab this tritium that moves over there, and this pair of electrons that were holding the tritium to this carbon actually come over here to form the bond to this one, and we get the three-membered ring that way. So the labeling experiments have shown something that's different. So that if I need to model this, I need to set up a reaction that can do this sort of stuff. Trying to come up with something with adding these nucleophiles to the system, I still have to figure out how I can do that in Wolfram language. Some molecules haven't been found at all. For example, this one here, nipetolactol, it's a 5-membered ring. This one is produced biologically, but my system didn't find it, and that's because it's produced in a very different way. It starts out with the same 10-carbon starting material, but it ends up also getting oxidized out here at the far end and here, and there's either a Diels-Alder reaction that forms the 6-membered ring here, that's this one, or it goes through a different route with getting the same final rearrangement. So if I were to include this in my system, I'd have to have some reactions that would be able to first do these oxidations and then allow these other additional types of reactions to occur. So overall, I do have a system now that I can start with the 10, 15 carbon precursor molecule, do these sorts of reactions and follow these rearrangements. I can drill down into the system and find out specific reactions that are used along the way from molecule A to molecule Z, et cetera. Other people have studied this as well already and published on it. The one that's closest to this work was published by Tian et al. They used essentially the same set of reactions we did, but they also considered geometry and stereochemistry and energetics. They did an exhaustive exploration. They didn't stop it at where things seemed to be reasonable. They also generated a lot of those 3-membered and 4-membered ring compounds in that first chart that were below the dashed line. They had custom Python code to do the generation, and they used the Schrodinger computational chemistry software to do all the energetics. We have some energetics and stuff built into Wolfram language. That's something that we could add on to this. De Silva studied sesquiterpenes, as did Isagawa, and they were interested in different things. So their methods are not quite the same as the ones we have here. So as I had said earlier, I'd like to implement more reactions so that we can get more of the observed biology modeled. I want to study the sesquiterpenes and the diterpenes also. And we also need to be able to consider stereochemistry, whether things attack a double bond from this side or that side, whether hydrogens end up pointing up or pointing down relative to the rest of the molecule. That's all important in terms of understanding the biological and chemical properties of the molecules. We do have the tools to do that. We just have to set them up, try them out, see how well they work. And I think that brings us to the end. The last thing I want to do is acknowledge the chemistry team, especially Jason Biggs. Without him, none of this stuff would work. He's the one that put together the original molecule implementation and all the code for molecule pattern, for pattern reaction and apply reaction. Jason Sonnenberg for a lot of discussions, keeping us on the straight and narrow in terms of molecular orbitals and energetics and things like that. Jan Mangelden has a lot of good insights as well. Bjorn and Michael Trott for their support for all this work. And also from the Fundamental Physics Project, I've made use of the token event graph. That was Nick Merzin's work. I've done some work with direct search with Brad Klee's depth first search function. So I can start out with a molecule and not actually have to build the graph itself, but I can still search that same space with a depth first search and go from one starting material and find a particular target, at least find one route there. So I'm doing that. James Boyd has been very helpful in terms of understanding things about multi-way systems and multi-computation. And of course, Stephen Wolfram for catalyzing all of this work and prodding me to get some of this chemistry stuff going under multi-way systems. And so that brings me to the end. Thank you for your attention. And you're only 10 minutes late for lunch. Thank you. All right, I'll take questions now as long as necessary. Yeah.
So it looks like a very natural follow-up. So it doesn't look like a big step forward to have to be able to actually put in reaction rates, forward and backward reaction rates for all of the edges in your network, and then automatically generate the differential equations that control the chemical.
Yes, we can do that. I have a Packlet in the Packlet repository, compartmental modeling, that'll let one do that. You can model epidemiology, you can model chemical reaction, systems biology. Yes, and I did not plant that question.
Okay.
Yes, we can do that.
Yeah. Okay, great.
Yeah, yep. That's why I wrote the package, because I wanted to be able to go from the reactions to the differential equations without mistakes, because I saw it too many times. People would add something, they would add a term to one reaction or to one equation and forget to subtract it or add it instead of subtracting it from the other one. And by writing everything in terms of transitions or reactions, and then from those generating the equations algorithmically, we avoid lots of those problems.
So are there plans to actually bring that Packlet into the mathematical system?
I don't know, that's not up to me, but it's there for you to use. It's in the Packlet repository. You can use it just by using resource function and the name of the function in my Packlet, or install the Packlet and just use it directly. Lots of documentation for it, including some systems biology showing chemical reactions.